So let's take a look at the details of Meiotic Prophase One. Now this is going to be the most complicated phase of meiosis we talk about. For our example, we are going to use a diploid cell with an N of 2, so a haploid number of 2. Now, when we're thinking about meiosis, we normally don't talk about the cell cycle. So we normally talk about the cell cycle when we talk about mitosis, right? So cell cycle goes something like this. So G1 to S to G2 to M, where M is for mitosis. I like to think, this isn't technically correct, but I like to think of meiosis being down here, like an alternative option. Now, cells don't do that. We have, let's say, humans, we have the germline, we have some cells that are destined to undergo meiosis. As far as I know, we don't know of any switch we can make or any chemical we can add and make a cell that was destined for mitosis to switch to meiosis. But I wanted to I like to think of it this way because the cell still needs to prepare for meiosis. And an important thing that occurs is that DNA replication must occur before meiosis begins. So we need to remember that the chromosomes have all undergone replication before meiosis begins. So they've undergone replication and the cell has prepared to undergo meiosis, similarly to how cells prepare. They replicate their DNA and they prepare for mitosis. So it's similar, and I like a diagram like this. It helps me remember that the DNA has already been replicated. So at the beginning of meiosis, do the chromosomes look like this here or this? So they're going to look like this because the DNA has already been replicated. Okay, so I just wanted to start with that. So when we're talking about meiosis, we need to remember that the DNA has already undergone DNA replication. So for a diploid cell with N equals 2, I'm going to diagram some chromosomes. So we have 2 pairs of homologous chromosomes. And the first subphase of Meiotic Prophase One is leptonema. So what happens during leptonema? Well, the chromosomes condense. So remember, before mitosis and meiosis, most of the chromosomes are not in a condensed form. They'd be very hard to see under a microscope. You really can't see them until they condense and you use a stain for DNA. So during leptonema, if we were to crack open a meiotic cell and look at it under the microscope and we didn't see the chromosomes or we just started to see them forming, we'd probably be pretty confident that that cell was in leptonema or leptotene stage. So I've already diagrammed them as condensed, so I will leave them like that. Another thing that happens during leptonema is a homology search begins. Now this is unique to meiosis. So this homology search, where homology sort of means similar, it's a similarity search, because meiosis needs to pair up the homologous chromosomes. So homologous chromosomes do not pair up during mitosis, but they must be paired during, did I say that right? Homologous chromosomes do not pair up during mitosis. They must be paired during meiosis. And the process that pairs the chromosomes is one of the least understood aspects of meiosis. After about 100 years of studying meiosis, with microscopes, and over 50 years using molecular biology techniques, we still don't understand how the homology search process works. But what it does, it will pair the homologous chromosomes together. So those two will be paired, and these two will be paired. This one down here will not be paired with that one. There's some sort of process that allows the cell to know when it has paired together homologous chromosomes and when it has tried to pair non-homologous chromosomes. So that homology search process begins during leptonema. So the next subphase is zygonema. So during zygonema, if we were to, look at some meiotic cells under a microscope and we saw that the chromosomes were roughly paired, so a rough pairing has occurred and they are about 300 nanometers apart, we would say that that meiotic cell was in zygonema. So let me diagram these here. So let's say this is 300 nanometers. And I forgot to diagram the nuclear membrane up there, so the nuclear membrane is still intact during the very beginning stages of Meiotic Prophase One. (300 nanometers). And what else happens during zygonema? A complex called the Synaptonemal Complex begins to form. So the Synaptonemal Complex is going to be a giant complex of proteins that forms along the homologous chromosomes, and it's going to link them together tightly. So it starts forming during zygonema. Now, at this stage, scientists typically refer to these roughly paired homologous chromosomes, right? There are 2 chromosomes in each pair, 4 chromatids in each pair. They usually refer to these as bivalents. The bi makes sense, right? Because there are 2 things. I'm not sure where the valence part comes from, but they call these bivalents. They're also called tetrads. So for our purposes, the term bivalent and tetrad means the same thing. Now, some people distinguish between the two because in many organisms, it's very hard to tell that each chromosome at this stage is actually made up of a pair of sister chromatids. The sister chromatids are so close together that the bivalents look like this. They're so close together that it's too hard to see that there's a sister there. So as we move through Meiotic Prophase One, those sisters become easier and easier to see, to the point where you can see them all, and then you have 4 chromatids, and that's where that term tetrad comes from. So the reason they're so hard to see is probably because cohesin is linked them together so tightly, and that cohesin is being degraded throughout between the sister chromatids throughout Meiotic Prophase One, everywhere except the centromere, just like in mitosis. And eventually it's very easy to see all the chromatids. And that's why, if scientists can see 4 chromatids in these pair of homologs, they call it a tetrad. If they can't, they call it a bivalent. But we're going to consider these the same thing. We'll assume that you can always see the sister chromatids throughout Meiotic Prophase One. So again, nuclear membrane is there. So I will diagram that there. So the next stage is pachynemia. So with this one, the synaptonemal complex is finished, is complete. Crossing over. Now we will talk a lot about crossing over. Crossing over occurs during pachynemia. And the homologs, which is the short name for homologous chromosome, homologs are very close together, 100 nanometers apart. So, quick diagram of that. It's going to look something like this. Probably should have diagrammed them closer. Okay, something like that. Now, I actually have them overlapping there, but so my goal is to show them very close together. And let's put that synaptonemal complex in. And crossing over occurs during pachynemia. And it's usually too hard to see that it has occurred because the homologs are too close together. So, nuclear membrane still intact. The next stage, now we're still talking about Meiotic Prophase One. We're going to go through the other Metaphase, Anaphase, all of those really quick because not much different happens there than the way those processes work in mitosis. So diplonema, what's going on? The synaptonemal complex is disassembled. Oops. Disassembled. So it disappears. And the chiasmata are visible. Now the singular form of chiasmata is chiasma. Now these are the points at where crossing over occurred. And what do they look like? So if we looked at a meiotic cell and we opened up that meiotic cell and stained the DNA and looked at it under a microscope, we should see the tetrads, or bivalents, same thing, looking something like this, held together by at least one chiasmata. So that's where the arms, at least one chromatid arm from each member of a homologous pair of chromosomes are touching, they're overlapping. So that's a chiasma. So every pair has to have at least one of those. If multiple crossing over events happen, there'll be two. There'll be two or more of those. And those are the points where crossing over occurred between the homologous chromosomes. And they're important because the synaptonemal complex has disassembled, so nothing's holding those homologous chromosomes together anymore. The only thing holding those together are the chiasmata. So they hold the chromosomes together and keep them from coming apart during Meiotic Prophase One. We don't want them to come apart until Meiotic Anaphase One. And why did the chiasma appear now? Well, because that synaptonemal complex has disassembled, and now the chromosomes can kind of move away a little bit. They can move apart a little bit. So it makes it possible to see the chiasmata. Nuclear membrane is still intact in diplonema. Okay, and for the final stage, diakinesis. Here, the nuclear membrane breaks down. NMBD, nuclear membrane breaks down. Kinetochores assemble on the centromeric regions. And just like in mitosis, centrosomes build or synthesize the microtubules, which attach to the kinetochores. And the microtubules push and pull the tetrads to the metaphase plate. Okay, so let me diagram that quickly while you think about that. So no nuclear membrane. Where did I put the chiasma for this other guy? Up here. So kinetochores. So each chromatid, here's a detail. Each chromatid will have a kinetochore assembled onto it at the centromeric region, but during meiotic prophase one, the two kinetochores on the same chromosome will act as a single unit. So that's a detail, which is kind of, I don't really understand exactly how the cell causes them to act as a single unit, but they do. Now, nuclear membrane has disassembled, so I won't diagram that, but I will show the microtubules extending from the centrosome regions. And each microtubule is going to attach to each kinetochore, and the chromosomes are going to be pushed to the metaphase plate. Okay, so that's it for meiotic prophase one. Let's talk about metaphase one next.